My name is Jordi Merino. I'm an associate professor and group leader here at CBMR, and it's my greatest pleasure to introduce Jose Florez. Jose is the physician in chief and chair of the Department of Medicine at Mass General Hospital. He's also professor of clinical medicine at Harvard Medical School and an institute member at the Broad Institute. Jose has been leading the discovery of genetic variants for type 2 diabetes and glycemic traits, and he has been leading global research consortia on discovering genetic variants associated with diabetes and glycemic traits, such as the MAGIC consortium or the NIH-funded Diabetes Prevention Program. Jose's work has been instrumental in understanding the pathophysiology of diabetes and how to use this information to advance pharmacogenomic approaches, and also how this knowledge can be implemented in clinical care, something that those days we call precision medicine. He has received many awards and recognitions. Maybe the most important was in 2010 as the Presidential Early Career Award for scientists and engineers. And this is the highest recognition level for early career scientists establishing their own career in research. So with this, welcome to Copenhagen, Jose, and looking forward to hearing you talk.
Thank you, Jordi. Thank you. Well, thank you very much for the very kind invitation to come here. Many of us feel that the CBMR is the mecca of metabolic investigation in Europe, and so it is really a pleasure and an honor to be part of this symposium. I have a very difficult task because I am the barrier between you and dinner, and it's been a long day of a lot of science, so hopefully this will be something that we can accomplish. It was right before the talk, I got a call from my daughter's school back in Boston. So that was kind of I'm a little nervous, but my wife has been able to sort out that it was just a congratulations because your daughter got admitted to the school. So everything is okay. So now all right. So over the last few years, we have presided over a major catastrophe. This is not an electoral map of the United States. Okay, this is how the obesity epidemic has been permeating across the decades in the United States. And of course, we know that right next on the heels of the obesity epidemic, there is type 2 diabetes. And so we have seen this happen before our very eyes. More people died from type 2 diabetes in 2021 than they died from COVID. 2021 was the height of the pandemic. More people died from diabetes worldwide from complications of type 2 diabetes than they did from COVID. So you see that this is permeating everything, unless you move to Colorado maybe, which is the one like maybe ray of hope in the country, then we are in trouble. And so then the question is, why is that? Because we're also in the midst of one of the largest therapeutic explosions in the treatment of this disease that we have witnessed. For many decades, all we had was metformin, insulin, and sulfonylureas. And as we all know, for the last 20 years, we have 13 different classes of different medications that are used to treat this disease. And yet, despite the very large and complex and very therapeutic armamentarium, this is where we are. So in data analyzed for the last 25 years, we see that the number of people who are at goal in terms of hemoglobin A1C in the United States is only about 50%. 50%. And at 8, for an hemoglobin A1C of 8, which is not great, but is more or less manageable, that was about a quarter of the population with type 2 diabetes. That is even worse. And this is despite, if you look, for example, at the number of people here in the center panel who receive at least across the various years, there's escalation of therapy. In red are the people who receive 3 medications as opposed to 2 or 1. So there's escalation of therapy. Medications are being added on top as we treat these people. And millions of people in one of the most developed countries in the world are still not at goal. And of course, the situation is similar worldwide. So why is this the problem? I think to a great extent, we still have a very incomplete understanding of pathophysiology, despite diabetes being one of the first hormone diseases described in human history, 1,500 years before the Common Era. We already hear the word diabetes. And despite insulin being the molecular form of therapy that addresses the deficiency in the disease, being available for over a century, thanks to work done by people that then became Lilly and Novo, creating that massive Despite having that available, we still have this limitation. So I have a complete understanding of pathophysiology. I will touch on why the therapies that we have to a large extent, they treat something that is not modifying the disease. We're not curing diabetes. I'm a clinician, and I have a diabetes clinic, and then people will come with type 2 diabetes to my clinic. And for the most part, unless they have bariatric surgery and maybe they get lucky with some of the new GLP-1s, I will have them as a patient for the rest of their lives. They will be my patient because I will not, as a clinician, be able to cure them, which is an experience very different from what my infectious disease colleagues have or even my oncologist colleagues have. So we don't modify the disease, we don't cure it, and then of course we have very ineffective public health strategies. So I think part of the problem is that we have defined the disease on the basis of an end result, and that is glucose. We define type 2 diabetes as hyperglycemia. Hyperglycemia that is not autoimmune and is not monogenic is called type 2 diabetes. I took this picture, so this is a candy store. I took this picture in New Orleans. It was the candy store that was at 7:30 in the morning. 7:30, it was open in the connector between our hotel and the American Diabetes Association conference meeting. So on our way to the diabetes meeting, this was already open, welcoming all of us. And because sugar is sort of at the center of this definition, and modifying sugar, because modifying the end result doesn't really change the disease course. It's sort of what we did for cancer 20 years ago. So the end result of cancer is growth and mass effect of that growth and adjacent vital structures. And that's how a cancer is discovered. And so we tried to apply therapies that simply targeted the end result, which was the growth. And so we have massive cell killing, nondescript, nonspecific radiation and chemotherapy that simply address the growth. And it wasn't until in cancer we were able to apply molecular therapies that attack the very target that causes abnormal cell proliferation, that we were able to make headways against cancer and actually cure it as opposed to palliate it. So I think what is needed really, when it comes to pathophysiology, beginning with, as Eli Zeghini just mentioned, having a catalog of the complete genetic architecture of type 2 diabetes. Of course, genetics is only half the story, maybe explains 30 to 40% of the variability in the disease. We need to understand how genes or alterations in alleles lead to difference in expression in specific tissues. So what is the molecular mechanism by which a nucleotide change leads to maybe a protein working differently or a gene being expressed differently, that then leads to something downstream that affects the cell, and then that in turn has an effect on the whole organism by physiology. What is the effect of perturbation, whether it's pharmacological or behavioral, on that abnormal molecular derangement? And then how do complications develop? So a deeper understanding of pathophysiology. I think parallel improvement and enhancement of therapeutics. So of course we need to lower glucose and bring glucose to the normal levels because we know hyperglycemia is deleterious long term. But then we need to preserve the beta cells, make sure that they survive and continue to make insulin through the lifetime of the person. We really don't have much in terms of ameliorating insulin resistance in target tissues. We need to have medications that are tolerable and people will take, and then that protect end organs. And then on the public health side, we need to be much better identifying who is at risk, and then how do we deploy an intervention that is effective? How do we get people to change the way they approach their disease and change their behavior in a way that is sustainable, not only in the developed world, but also in lower and middle income countries, and of course, something that is culturally sensitive to the context where people live. And so our research program in Boston is really trying to tackle all of these things, and I think one leads to the other and to the other. And so what I will tell you about in the next few vignettes in this talk is to see how, beginning with genetics, we can try to influence some of these aspects and hopefully beginning to make a headway that we will see genetics permeate the clinical space. So Eli talked about this. I'm not going to belabor this point, but basically over the last 20 years or so, there's been an explosion of genetics by which we now have hundreds of loci that explain a tiny effect on genetic risk. So as the previous talk from Dr. Radek mentioned, it is not just about any one gene. It's about a composite of many different genes, each with very small effect, working in concert with the environment to then raise the risk of diabetes. What's shown in this slide is by year of discovery, beginning in 2000 all the way to 2020, and then the odds ratio on the y-axis, meaning the effect of that allele on overall risk. And then the color denotes the mode of discovery. So initially, it was candidate gene association studies in green, a linkage study in purple, GWASs in blue, and then more recently sequencing experiments, which together have led to an explanation of about 50% of the heritable risk of diabetes, about half the way there on cataloging and unearthing the entire genetic architecture of type 2 diabetes. And I think here it's important to sort of step back a little bit about scientific discovery. And I'm going to get a little philosophical. I think when it comes to human history, political history, sometimes it seems that we go in cycles, that we repeat history over and over again. We fight one war, and then some years go and we fight the same war, and then here we are again, another European war. Or we elect a group of people, and then the cycle comes down, and then we elect another group of people, and we don't seem to sort of learn, and we are sort of in this loop. But when it comes to science and discovery, and I take comfort from this, it's incremental. There is a time when finally a discovery gets made, and there is no going back. Something has changed, and humanity will be different after that has happened. So I think, for example, in my own institution at Mass General in Boston, we discovered the use of ether for anesthesia, to eliminate pain. And there was a world before anesthesia where having a toothache and going to the dentist was torture, or where you had to deliver babies without any ability to have that pain taken away. And then there is a world after anesthesia. And the world changed not too long ago, 100 years, 150 years ago. The world changed because anesthesia was there. The same thing can be said of antibiotics. An infection, only in your grandparents' lifetime, an infection could be lethal. We just heard about this person who died from pneumonia at age 41 and could have changed the face of genetics and had a simple pneumonia that none of us would think twice about, just taking a pill for a week and be healed from, and this person died in 1902 or in the early 1900s, so not that long ago. So there's a before and an after. I will say that there's a before the genome and after the genome. And so all of you here, I think, hopefully, well, maybe some of you are, the genome was sequenced in 2003. Anybody born after 2003? Okay, so all of you here were alive when the human genome was sequenced. Very soon I'm going to have to stop saying that. But at this time, everybody in this room was born when that happened. And so you are living in an amazingly exciting era where now we have the entire blueprint of the human condition at our fingertips. And then the question that many of us are engaged in is to see whether that information will transform or not transform the practice of medicine. And there are some places where it will and some places where it might not. And I will try to show you that when it comes to type 2 diabetes, I think in some instances it will. So the question, can human genetics help understand the heterogeneity of what we call type 2 diabetes, stratify the population to make reasonable decisions, implicate new metabolic pathways that might help us understand and potentially target for therapeutic discovery, and then clarify in which direction altering that molecule, that gene, that transcript has an effect on disease, hopefully establish what is causal versus what is a consequence, and then explore off-target consequences. So for the rest of the talk, I will just give you one or two vignettes about each one of these points. So about understanding the heterogeneity of disease, you're a clinician, a general practitioner, and somebody comes to your clinic and they have hyperglycemia, you say they have diabetes, and the main decision you need to make is this type 1 or type 2, because we know that type 1, which is the autoimmune destruction of beta cells, requires insulin therapy, whereas type 2 can be treated in many different ways. Now, if you're an endocrinologist, you probably have a few other syndromes that you can catalog that go beyond type 1 and type 2. And you know that besides those two divisions, there is also something called, for example, LADA, which is latent autoimmune diabetes in adults, which is a form of autoimmune diabetes that happens later in life. You have MODY diabetes, which is monogenic diabetes. And there's a number of things that you can define, ketosis-prone diabetes. But then the rest of it you call type 2. As I said earlier, anything that is not autoimmune or monogenic, you lump in this big basket and you call that type 2 diabetes. But we all know from clinical practice that type 2 diabetes presents in many different flavors. You have people who are lean and get hyperglycemia later in life, and some people are obese, and some people have a pregnancy, and then after that they develop diabetes. There's a number of things that are really heterogeneous. And so this grab basket that we define by hyperglycemia that is not autoimmune or monogenic, which we call type 2, is really heterogeneous and probably has many different pathophysiological ways or mechanisms by which people become hyperglycemic. And so when you try to apply genetics to something like this, if I can get the slides to move. Okay, so back and forth. There you go. You can do, for example, a GWAS. And now what we have here is the same Manhattan plot that Elias Guini showed earlier, but now it's turned 90 degrees. So what you have is on the y-axis is all the chromosomes, and then on the x-axis is you have the effect size, and each of the dots is a particular genetic variant. So you do a GWAS, and then you can, instead of measuring the tiny effect of each one variant on diabetes risk, you can do an aggregate and construct what we call a polygenic score, where you take the entire aggregate of all the effect sizes, and then that is your overall risk of diabetes. And the way you collect those variants can be, well, let me just do what I call restricted to significant polygenic score. That's a score that only gathers the variants that surpass the very stringent threshold of genome-wide statistical significance. So you need to get to 10 to the minus 8 as the p-value. And then those are the ones you collect into your type 2 diabetes polygenic score, maybe a few hundred variants, and then you construct a score in that manner, which explains some proportion of the variants in the disease. Or you can take, actually, you can say, well, there's going to be a lot of false negatives under the top very stringent statistical threshold, where there's many variants that are actually causal for the disease, but haven't really risen to statistical significance. So let me construct what I will call a global extended polygenic score. And again, you now have many more variants, and with that, you might be able to explain a higher proportion of the disease. Neither one of the two approaches tell you much about mechanism. They tell you about overall aggregate risk of getting hyperglycemia that is not autoimmune or polygenic or autoimmune or monogenic, but it doesn't tell you much about mechanism. So what Miriam Udler in our group has been able to develop is what we call partition polygenic scores. And what she does here is, yes, she takes all of the variants, but then has the phenotypes of the people, the endophenotypes, for example, glucose levels, LDL cholesterol, body mass index, insulin levels, has that drive how those variants are grouped into what we call partition or process-specific polygenic scores, like by a clustering mechanism. And by doing this clustering method, what she's able to show then is in the initial paper, which has then refined in 2 other iterations, is that there's at least 5 different genetically defined, genetically anchored, but physiologically informed clusters, 3 of which have to do with insulin resistance and 2 of which have to do with beta cell function or processing. So there's an insulin processing and secretion cluster, there's an insulin synthesis pathway. These are defined by lower insulin levels, and then 3 that have higher insulin levels, and therefore insulin resistance, a lipodystrophy-like cluster, obesity cluster, and a liver insulin resistant cluster. Now she's taken that with more GWAS findings, higher cohorts, more phenotypes, and now we are at about 11 or so different classes that are really refined. And these figures, the way they are arranged, just to guide you, and I'll tell you one example where this has been very informative, is you have these circles, and then around these circles, in the blue, for example, you have traits that go down for people in the cluster. So in this case, maybe insulin secretion. In the red are the traits that go up, and in the green are the genetic variants that define the cluster. So here you have 11 different clusters. And what we've been able to do here, for example, is to explain the difference in phenotype for type 2 diabetes between people of East Asian descent and people of European descent. Because when you focus on lipodystrophy and liver insulin resistance as the subtypes of type 2 diabetes that work via insulin resistance, but without having an effect on BMI, so they are insulin-resistant people with normal body mass index, and you see what the distribution is of those 2 clusters in various populations. So in green is Africans, in yellow is Americans, East Asians are in blue. You see that the East Asian distribution for both the lipodystrophy cluster and the liver insulin resistant cluster differs from the other groups. They have a much higher genetic prevalence. If you're of East Asian descent, you have a much higher genetic prevalence of these 2 particular subtypes of type 2 diabetes. And so when you then examine in the graphs below at various BMI indices, people of European descent in the red get diabetes risk at this BMI of 30, and you compare what happens, what is the similar prevalence in East Asians, and you have East Asians getting type 2 diabetes at a BMI of 24, average BMI of 24. So there's a 6-point difference in the risk of obesity conferring diabetes on people of East Asian descent. And you ask, why is that? We've never been able to explain that. Why do people of East Asian descent get diabetes at a lower BMI? It turns out that the difference in prevalence of the genetics for these 2 clusters explains as many as 63% of that difference. So in the graph on the right, you see that the blue curve, which is where the East Asians are now a lot closer once you adjust for the effect of this genetic background on that. So that tells you a little bit about the subtype of diabetes that happens to be more prevalent in the East Asian continent, explaining something that we didn't understand until this work was done. So what was try to find the population? So here the thought is, can we then use genetics to be able to say we're going to treat people differently? And the very first inkling that this might be helpful would be in work that we did very early on in the diabetes prevention program. So this was a clinical trial funded in the United States with people with impaired glucose tolerance, so a high risk of developing diabetes. And then they were randomized to either placebo standard of care, which is some classes and go off and see what happens to you, and then a very intense lifestyle modification with diet and exercise, or metformin. There's a fourth arm, triglyceride, that did not finish, and we'll concentrate on those three. They were followed for 4 years, and then DNA was collected, and everybody knows that both the metformin intervention and the lifestyle intervention were quite effective at delaying, or at least preventing the onset of type 2 diabetes, with a 58% risk reduction for lifestyle and 31% for metformin. So on that cohort of people, we had genetics, and at that point, the very first common gene variant for type 2 diabetes had been discovered by the Decode Genetics group in TCF7L2. So we asked the question, what happens when you carry the risk genotype at TCF7L2 and you're enrolled in the diabetes prevention program? Do any of these interventions work differently depending on your genetic background? And so 10% of the population, so I don't know, 50 people maybe in this room, have the TT genotype, which is they're homozygous for the risk allele, the TT genotype. And if you're on the placebo arm in the diabetes prevention program and you happen to have this genotype of this one variant where you have both alleles of risk, then you have an 80% higher risk of developing diabetes. And this is above and beyond already having impaired glucose tolerance and having a high BMI. So over that, you get 80% additional risk from having both copies of this allele. But if you're in the lifestyle intervention, it turns out that that risk goes down to baseline. That risk goes away. And this makes the point that Dr. Radek was making in the previous talk, is that environment matters. And so as opposed to genetics being deterministic, as in Mendelian genetics, where if you have the cystic fibrosis gene mutation, or if you have the sickle cell mutation, then you're very, very likely to get the diabetes because of the high penetrance. In this case, environment matters. What you do with diet, what you do with exercise actually abolishes the bad genetic risk, the inheritance that you receive from your parents, to bring that risk to baseline. So you will say, well, maybe that's just one variant. And so Marie-France in our group decided to then take that and do a polygenic score, as I mentioned, at that point grouping all of the genetic variants that were known at the time and deriving a polygenic score, and then dividing people by genetic risk quartile, with the lowest genetic risk on the left, the highest genetic risk on the right, and then evaluating placebo versus lifestyle. You see that if you are in the placebo arm, your risk of diabetes goes up the more genetics you carry. And then you see that the lifestyle intervention is really, really good at bringing that risk down to baseline, particularly effective in the highest genetic risk. And when it comes to stratifying the population, and if you don't have the resources as a society or as a country to give everyone in your population at risk the lifestyle intervention with very intense exercise, a trainer, a dietitian, maybe you can do that for the people at the highest quartile of genetic risk, because for those people, it really matters. And in work that is not yet published, but Jordi Merino has been doing here with some of his group, there's already some hints that this very high impact of activity, physical activity, on a particular quartile or distribution of genetic risk is effective, and there seems to be a positive interaction. So it's confirmed by work that is happening right here. What about implicating new metabolic pathways? Are we understanding more about the disease as a result of this work? So I'll tell you a little bit about the study to understand the genetics of the acute response to metformin and glipizide in humans, which is spelled sugar and GH. And what we try to do is, can we take human participants and give them two different pharmacological perturbations and then study the genetic response to that? So we wanted to assay insulin secretion, and for that we gave people glipizide, which of course stimulates insulin production in a glucose-independent manner. And then we wanted to assess hepatic glucose output, and for that they received metformin. So in this protocol, all participants, about 1,000 of them, who didn't have diabetes, received both interventions sequentially, and then we have DNA on both of them, and we did a no GTT. And so nothing really was known. I'll tell you about TCF7L2, it's a transcription factor. It had been discovered, but no one really knew about how that particular genetic variation, that gene, that protein, led to type 2 diabetes risk. We were able to show in this cohort of sugar and GH that when you measure GLP-1, whether it's total or active, in an OGTT, after you receive metformin, and you stratify people by genotype, it turns out that people who carry the risk genotype, the TTs that we saw earlier in the diabetes prevention program, you're homozygous for the risk allele, you make more GLP-1, and yet you have higher glucose, and you have lower insulin. So that tells you that you have more GLP-1, but less of the downstream expected effects of GLP-1, which really infers GLP-1 resistance. There is incretin resistance. For some reason, having this genotype leads people to respond less well to endogenous GLP-1 production after a meal as a potential mechanism of action. The other example came from work we did in a Mexican cohort, so the Sigma Consortium. This was a GWAS of 9,000 people from Latin American descent. We did this GWAS with the understanding that genetic variation that had not been assayed in Europeans was there to be discovered, and other groups also need to be studied. And when we did this big GWAS, we found a variant in this gene called SLC16A11, where the allele is very common in people of Latino descent. So you will see that it's about 50% prevalence in people of Native American descent and less than 2% in European descent. So it turns out the variant arose in Neanderthal populations. So I don't know if you know this, but Neanderthals and modern humans coexisted in Europe, and then it got introgressed into modern humans by contact with the Neanderthal populations, and then that allele traveled eastward. So it's about 11% prevalent in Eastern Asia, and then with a crossing of the Bering Straits, it's a bottleneck. So only a handful of people made it across, and it's completely stochastic as to how many people made it across. And apparently families that carried the variant were the ones that made it, and so it really spread in the Americas. So we're able to discover this in Europeans because in Mexicans because it was much more frequent. So we have many more observations to be able to reach statistical significance. And so then the question is that what happens, how does this work in terms of mechanism? And also, how does this work in terms of advancing the slide changer? Okay, there we go. And so it turns out that the variants that we discovered both coding and non-coding. So coding means that they change amino acid sequence, and then non-coding, it changes the regulatory sequence. And both of them have similar effects. So under normal conditions in the wild type, SLC1611 is a monocarboxylate transporter. So it's a molecule that is in the plasma membrane, and it allows small ions like lactates or acetate to get across the membrane. And so to get to the membrane, it needs to be expressed. And then there's a chaperone called basogen that carries it to the membrane and makes sure that it gets where it needs to be, and it's mostly expressed in hepatocytes. And so if you happen to have the coding variants that change amino acid sequence and that give you high risk of diabetes, that interferes with interaction with the chaperone. So you really don't bind basogen so well, so it interferes with the transfer. And if you carry the regulatory variants, it interferes with expression. So it turns out that people who carry this haplotype, meaning the linear sequence of alleles that give you higher risk, both coding and non-coding, you have about a 90% decrease in the expression of the transporter on the membrane. And the other twist is that hepatocytes are not the same. So the way the liver is organized, you have a central vein that is very low in oxygen that drains the liver, and then you have a portal vein, hepatic artery, that is on the other side of this sort of triad. And so there's a high oxygen gradient at this type of hepatocytes that live near these two vessels, and then a very low oxygen tension hepatocytes that live over here. And if you look at all the genes in the liver that are expressed in human or mouse hepatocytes across this variant, one of the genes that has the strongest difference between high oxygen and low oxygen is SLC16A11, going to the point that you can't really do liver homogeneous to try to study this. You need to go single cell. And then what we see is that if you carry the variant and you don't have enough levels of SLC16A11, you don't get rid of lactate. So lactate accumulates inside the hepatocyte. And then that leads to downstream consequences in terms of lipid accumulation and other deleterious effects to the hepatocyte that makes the liver less efficient at trying to do what it does, for example, for glucose. What this allowed us to do is that by not only identifying genetic variants that have an effect on a human phenotype, but also being able to say, because of the change in expression, we can tell the direction of effect, meaning that the effect of this genetic variance is loss of function, lower levels, then we actually have a direction in which we would approach therapeutics. What is needed is to raise levels of SLC16A11 in the plasma membrane, a feat that is not impossible if you look at how cystic fibrosis is treated today, where you actually have small molecules that are able to modify the ability of the chloride transporter to regain the conformation that allows it to function at the membrane, and then another small molecule that is able to help carry it to the plasma membrane. So not something that is science fiction. What about identifying potential molecular targets? So a lot of people say, well, you spend all this money on this investment in GWAS, and you have all these lists of various genes, and then, really, how has it changed medicine? And some people are skeptical about this. I think I'm very, very grateful that Jim was able to show the trajectory of GLP-1. In 1987 is when GLP-1 is discovered, also at Mass General. And I think the first approval for a molecule is in the mid-2000s. for a diabetes indication, and then we really didn't have a weight indication until much more recently. And so, it's taken 30 to 40 years to get to this point. So, GWAS are only about 20 years old, so give us some time, and then we'll be able to say. But what I will tell you, when you take all the GWAS discoveries shown here in this very nice circle from one of the more recent publications, buried in that list is PPR gamma as a gene that, when modified, gives you high risk of type 2 diabetes. And of course, PPR gamma is the target for a class of drugs that has been used to treat type 2 diabetes and has a clinically significant A1C reduction. Now, there's other issues with these molecules, but it has been approved, and it has a clinically significant A1C reduction buried in that list. Also buried in that list is a target for sulfonylureas that also a very small effect on diabetes risk, but is present in that list as a gene that when targeted with a medication, you might reduce A1C. And the same thing for GLP-1 receptors, one of the things that arises. So what that tells you is that GWAS give you a list of 700 genes, and buried among them, because there's some that are true positives, there may be others that could also be, we identify them and we really have a good sense of what is the effector transcript that leads to the change, may be used for therapy. And if you go to the cholesterol field, it's even more impressive. Buried in the GWASs for LDL cholesterol is HMG-CoA reductase, and of course that molecule, when targeted, not only lowers LDL cholesterol but also saves lives. So the point here is that there may be things that we can try to pursue. So one example is in this GWAS that was done for metformin. We really don't know how metformin works, and then Ewan Pearson in Dundee with a consortium did a GWAS for metformin response, cut to the chase, and then identify the GLUT2 transporter, that transports glucose, mostly expressed in human liver, but also in human beta cells. And in this regional plot, you see that the genetic variant that rises to high significance is right here in SLC2A2, which is the gene that encodes the GLUT2 transporter. And then the very same variant is co-localized, or also has an effect on the expression of the transporter in human liver. So the very same allele that leads to better metformin response also leads to higher expression of the transporter. So that was known about the GLUT2 transporter and metformin, but now we're doing the genetics, and this is unpublished work on GRADE. GRADE is a clinical trial funded by the NIH, conducted in the United States with about 5,000 participants that randomize people, these 5,000 participants, to 4 different groups. So they're all on metformin to begin with, and decent control, and then they randomized to either sulfonylurea, glimepiride, a DPP-4 inhibitor, sitagliptin, liraglutide as your GLP-1 analog, and insulin. And so the clinical results from this trial were published in the New England Journal a couple years ago, and it showed, in terms of the primary outcome, which is failure of therapy on top of metformin and also A1C, this is how they do, and of course, liraglutide and glargine had the strongest effects, although, it was clear over time they were better. So we're doing the genetics on this, and again, this is not published, and lo and behold, when we look in the glimepiride arm, the one variant that comes out is from the same GLUT2 transporter. And it's a variant that is very strongly correlated with the one that had been discovered in the metformin GWAS. So we have the very same variant in a GLUT2 transporter that is mostly expressed in liver, but also in human beta cells, that directs or determines response to metformin and glimepiride, but what's really critical here is that it's in opposite directions. So the allele that makes you a better responder to metformin is the same allele that makes you a worse responder to glimepiride. And we've done some functional experiments in zebrafish that seem to try that recapitulate what we see in humans in terms of, because we understand the level of expression. And so now that really gives you some anchor when you're trying to make a decision, particularly in countries where metformin and sulfonylureas remain the workhorse of type 2 diabetes therapy, which is in lower middle income countries, in Africa and some places in Asia, then it actually makes a difference whether you prescribe one or the other based on your allelic variant that you carry. Let's skip that. Okay, how about clarifying the direction of effect? So this is an interesting story, and if you are, for people who are involved in pharmaceutical companies, I think it's illustrative. So you may remember that the very first GWAS for type 2 diabetes was reported in Elsinore. So just a few kilometers from here in Elsinore. In 2007, there was a conference, and Philippe Forgel was at that conference, and then he reported the very first GWAS for type 2 diabetes. And TCF7L2 was one of the things that came out, but as a novel association was this zinc transporter, which is encoded by SLC38. And it encodes a zinc transporter called ZNT8. Now, Turns out that ZNT8 was already known. ZNT8 is a zinc transporter that is co-expressed with insulin. So you see here in beta cells, when you stain them for ZNT8, you see this granular pattern in islets that is mostly in the periphery of the beta cell. Granular because it's sort of the cytosolic in the granules and it's co-expressed with insulin. It's a zinc transporter that brings zinc into the insulin storing granules, and that's how insulin is made stabilized in granules before they're secreted. And this had been shown about 2 years before the GWAS. So you could say, well, why did you need a GWAS to be able to do this very expensive GWAS and thousands of people when you already had this finding? Was it just simply confirming something we already knew? We may have known that a zinc transporter existed that was helpful for zinc to be transported into granules to store insulin. What we didn't know is that the natural experiment by which variation in the zinc transporter, coding variation, where an arginine becomes a tryptophan at position 325, that is enough to give you a phenotype in humans. So very different, it's not only knowing that something exists that sits in a beta cell, is that when you tweak it, when you perturb it, in this case genetically, you can actually affect a phenotype in humans. So GWAS gives you that information that you didn't have before when you simply did the descriptive biology. But on the basis of that finding, that an arginine becomes a tryptophan and that gives you a higher risk of diabetes, there were drug companies that launched research programs to try to modify this as a target and see whether we got a phenotype in humans. And they assumed that because they're having an amino acid change, and that was something that seems to be good, transport zinc into granules, and you need zinc in the granules, then maybe the variant, what it's doing is it's actually interfering with doing something. Like what you need to do is you need to do an activator. It's interfering with the function of the channel or the transporter, and you need to design an activator that is going to make the transporter work better. That was the assumption. And so programs were launched in drug companies to do that. And then the question is, well, did the genetics really tell you what the direction of effect was? Was this change from arginine to tryptophan gain of function or loss of function? That wasn't really known. And so genetics can tell you that because what you can do, maybe an amino acid change doesn't give you the answer whether it's gain of function or loss of function. You really should not launch a research program before you answer that question. But if you do sequencing in enough people, in this case 150,000 people, and you look for protein truncating variants, so stop codons or splice junction that makes you lose an exon or things of that sort, then you end up with protein truncating variants that really you're sure that produce loss of function. And then you ask, what is the effect on people when that happens? And what was shown is that loss of function mutations of this gene are enriched in people without type 2 diabetes. Loss of function is protective. So designing an activator is the wrong thing. What you need is an inhibitor. An inhibitor of the transporter is what's going to give you the therapeutic effects that you seek. And then a follow-up paper in many, many more people, now hundreds of thousands, kind of confirmed that a lot of the loss of function variants were overrepresented in people without diabetes that were protective. And so then that was followed by an entire research program, much of it conducted in Lund University, just across the bridge, where using both humans and cells and animal models, all of that really converged on that answer, which is when you look at insulin secretion, showing in one of these graphs here for humans, that insulin secretion is improved when you actually inhibit or have either genetic or small molecule modifiers that give you to loss of function in the transporter. So direction of effect. Getting to the end, establishing causal inference. Of course, a lot of epidemiology is observational and a lot of it is correlational. And so you want to know what is cause and what is effect. Can I prove that something intervening on something is going to have an effect? And many times to do that, you need to design a randomized clinical trial, which is an inordinately expensive and laborious process that takes many years to complete, depending on the outcomes that you select and the strength of your intervention. And so are there shortcuts that one can take to try to establish causal inference without having to spend all that money for some of the questions? And can genetics help in that matter? And so here I want to just touch on something that Jordi Marino did when he was a postdoc in my group. And so one thing that was known for a long time is that there's a correlation between hyperglycemia and coronary disease. So in clinical practice, we are told to treat diabetes as a coronary disease equivalent. So in terms of risk factors assessment, if you have diabetes, it's as if you had smoking, hypertension, high cholesterol, etc. So they travel together. Hyperglycemia and coronary disease happens. And yet, when the initial trials in the early or the mid-2000s So ACCORD and ADVANCE and VADT, 3 major clinical trials that ask the question, does intervening in type 2 diabetes to lower glycemia have an effect on coronary disease? Those trials were negative, which was very bad for endocrinologists, because we think that lowering glucose is good. We had seen that in type 1 diabetes. We had seen that for microvascular complications. We really wanted to see it for macrovascular. The UK-PDS trial that tried to answer that question came to a p-value of 0.052. So they could not really claim that it was significant. And ACCORD and ADVANCE and VADT were supposed to give us the answer, and they didn't. Now, when you meta-analyze them, or when you look at the long-term follow-up of the UK-PDS, it looks like actually it does matter. But there was this controversy at the time between cardiologists and endocrinologists as to whether intensive glycemic lowering was helpful. And this is before all the cardiovascular outcome trials that came with SGLT2s and GLP-1s. And so then Jordi says, well, can we use genetics to answer that question? And the one interesting thing about the genetics of hyperglycemia is that when you look at a GWAS for glucose levels, not type 2 diabetes, glucose levels in non-diabetic individuals, there was this odd phenomenon by which in the purple are genes that increase glucose and also affect diabetes risk. So they are the same ones that show in GWAS for type 2 diabetes as a disease phenotype. But there are some in green that seem to have a big effect on glucose, and yet they don't really have an effect on diabetes. They seem to move glucose within the non-diabetic range, but they're really not giving you a high risk of diabetes, meaning that you can genetically create a polygenic score that separates hyperglycemia from type 2 diabetes. Because you don't want the other things that go along with type 2 diabetes, like hypertension and lipids and obesity, contaminating your assessment of whether simple glycemic exposure leads to high coronary disease. And so then you can implement something called Mendelian randomization. You have an outcome, in this case is coronary disease, heart attack. You have an exposure, a risk factor, in this case is glucose, and you don't know which one is causing which. It could be that somebody has high glucose because they're having the heart attack and they get all nervous and they have all these things and then glucose goes up, or glucose is the cause of the heart attack. There may be confounding factors. There may be things like obesity that leads to both high glucose and heart attack, and so both are happening at the same time because of something that is confounding. But if you can identify an instrument, in this case a gene, that predates one of the exposure and is intimately tied to the exposure because it has a direct effect on the exposure based on the biology, and then you show that that instrument has an effect on the outcome, then you've established a direction of effect. That's called Mendelian randomization. So the randomization is not participants coming and you randomize interventions. The randomization occurs at meiosis, when your gametes, your alleles are randomized, and the allele that gives you high glucose is stochastically assigned to you versus the allele that gives you low glucose. And so what you have is genetic exposure to glucose as your intervention, your exposure, and then you see, does that have an effect on heart attack? And so what Jordi showed is that for every millimole per liter increase in fasting glucose, you have a 43% higher odds of having coronary disease. And if you refine your instrument to eliminate some of the variants that may have had an effect on lipids or body mass index or blood pressure, you still have a 33% higher risk of coronary disease for every millimole per liter increase in glucose, for the first time showing that hyperglycemia as a genetic exposure puts you on the path to heart attack, and intimating that lowering glycemia then would be beneficial. And only 2 years later, EMPA-REG was the very first cardiovascular outcome trial using SGLT2 inhibitors, where you saw a significant, it was only shown for safety, but actually for non-inferiority, but it showed superiority. It showed that EMPA-REG, as an SGLT2 inhibitor modifying glucose levels and other things, was able to lower risk of heart attack. And of course, now we have all the GLP-1 literature. And I will close with how you can use genetics also in a pharmaceutical company setting to explore off-target consequence. You may have a molecule, you may have your effect. You've only seen people during the course of your trials, which is a limited exposure, maybe a year, maybe a few years. You don't know whether you're going to have off-target effects that could be really problematic. And your phase 1 and your phase 2 studies are too small to really see what happens in the population. The way that's dealt with today is you have a phase 4 sort of post-marketing surveillance study, and you see that when you deploy it across the entire market of population, hopefully you don't have a signal that makes you worried, and of course that can happen. Well, one way to try to do that, you can do that genetically as well, because you can do what's called a PheWAS, or a phenotype-wide association study, where you see, does this genetic variant that causes that effect on my exposure, does that lead to other things besides the one outcome that you want to modify? And so this was done by a medical student in our group trying to answer the question, do sulfonylureas lead to higher coronary disease risk? That had been something that had been suggested, and many people do not prescribe sulfonylureas because they thought it had a bad effect on coronary disease. Turns out, as I mentioned earlier, KCNG11 and the sulfonylurea receptor, we have genetic variants that serve as the instrument that basically recapitulate the effect of the drug. And so when you ask, do people who carry those variants genetically, do they have a high risk of heart attack? The answer is no. There's no increased odds ratio, as you see here in this meta-analysis, for people who have that. And then on the heels of that study, which basically took a medical student 3 months with a database to answer the question, having all the math and very quickly being it done, in the UK Biobank, there were 2 major clinical trials costing millions and millions of dollars over 7 years, showing that sulfonylureas were not harmful when it comes to coronary disease when compared to the gliptins in this case. So the answer really is that human genetics can help in ways that benefit the drug discovery, that identify potential targets, that serve to stratify the population, and that will need to be deployed to assess polygenic risk in a way that helps us characterize the heterogeneity of disease. And so the idea here, as I was mentioned by Dr. Radek earlier, is that it's not just genes, it's not just the way those genetic variants in the genome affect expression or the epigenome, the transcription, how they affect the proteins that those genes encode, the metabolites that those enzymes generate, all the different environmental exposure of the lifetime of a person. It's a combination of things that all need to be harnessed to be able to predict risk. And so what I envision is that genetics can be as a skeleton, the anchor. It's stable, it doesn't change. Germline genetics is the same in the life of an individual. It can be measured once in your lifetime at birth. And then you have your genetics as a skeleton that only explains about 30% of your overall risk. And so on that, you have to superimpose environmental modifiers. The analogy here is the anatomy. That would be kind of muscle that makes you strong, the activity, etc. Circulating biomarkers that may give you a sense in time of what is the metabolic state of the person as you proceed through life. And then behavioral traits in terms of how you act with diet and how you act with your physical activity. And as the composite of all of those, that then gives you a holistic way of predicting whether you have risk or not and how you might respond to medications or develop particular complications. So with that, I just want to conclude thanking many people in the group over the years, the many consortia that I've been very fortunate to work with, funding agencies, and then the people at home supporting this thing. And this is the one that I thought I was really worried about, but the phone call was about her really making it into the next phase of her schooling and not having a, some sort of major bleed. So thank you very much. I'm happy to answer questions.
Amazing lecture, Jose. Thank you so much. So now we will have 10, 15 minutes for questions. Are there any questions? I think over there.
Thank you for a wonderful talk. That was really great. I had a question to the last part of your talk, because first you showed that actually it did make sense to lower blood glucose in terms of preventing cardiovascular diseases. At least that's what I understood. And then the next thing is we looked at the sulfonylurea that did not have any effect on the cardiovascular outcome, but I would assume they still have lower blood glucose. So how is that explained?
Oh, that's a good question. So the effect of the single variant of the sulfonylurea is very, very small. And the key thing there is to show that it was not harmful. So we had enough power to show that it wasn't harmful, and that's what was shown in the other clinical trials as well. The reason that particular variant was not included in the polygenic score because it has an effect on both diabetes and glucose. It was not just an isolated glucose variant. But yeah, you're right. I mean, I think if that variant had sufficient effect, then you should have seen a benefit. Thank you.
Congratulations, very nice talk, even for me that I'm not a genetist. I was wondering if you ever look at the genes that are now recognized for muscle DMH, because these are really interesting because, of course, they predict the disease, but on the other side, they're not related to diabetes or cardiovascular disease. Actually, some are even protective because they are associated with lipoprotein secretion. So since they block the lipoprotein secretion or they impair lipoprotein secretion, they have a better cardiovascular outcome. So it's sort of a dichotomous because they do not act, they act only on the liver disease, and they do not increase glucose production and hyperglycemia, not probably only in some subjects, but are not really associated, and they are protective on cardiovascular disease.
Yeah, so that's a great question. So when we look at all the genetic variants that influence risk of type 2 diabetes, and then we do the clustering work and see how do they cluster, and we took many traits to do the clustering, including liver enzyme levels. It turns out that one of the clusters that emerges is a liver insulin resistant cluster. And then you zero in and you find out, okay, what are the genes that are really showing up in this cluster that the hallmarks are higher levels of insulin resistance, normal BMI, and lower triglycerides, actually. It turns out that some of those genes are also coming up in GWASs for MASLD. And so there is a genetic overlap correlation between a form of diabetes that seems to be focused on the liver and on the ability of the liver, for example, to process and secrete triglycerides that lead to fat accumulation in the liver, steatosis, steatohepatitis, and metabolic disease from that. So there's definitely something there that you've pointed out.
Hi, Alvin from CBMR.
Yes.
Given that RCTs remain the gold standard for driving clinical practice, but haven't yet to fully integrate precision medicine or genetic stratification, how do you envision complex genetics influencing treatment guidelines for type 2 diabetes without all this precision medicine-driven RCT? Or do we have to wait for a precision-driven RCT?
Well, that's an excellent question. So there's a number of things to your question. I think the first piece is that there is never going to be a way in which de novo genetic testing will be part of a clinical indication. That is unaffordable. You cannot be that, okay, I want to do this and I'm going to do a genetic test for this person to see whether I make a clinical decision. You take out all the phenotypes and all the drugs, and there's no way that as a society we can afford that. But I think what's going to happen, because germline genetics is very cheap to obtain, you can do for $50, you can do a genome-wide genotyping, and sequencing costs continue to drop, and you only need to do it in the lifetime of an individual. I don't think it's unreasonable to expect that fairly soon, at least in the developed world, that your entire genetic sequence will be part of your medical record, right? So people will be born, and then they'll be sequenced, and then that gets stored. And then that information then can be mined whenever there is a clinical indication that emerges at no additional cost, right? So you have to take the cost of the genetic testing out of the equation, and I think that's not unreasonable to expect that that will happen. So then what we need to prove is what you suggest, is that we need to show with our work that having that genetic information leads to a clinical decision-making that makes a difference, that changes outcomes. And so that's the evidence that we need to gather. So when that happens, I think the idea would be that somebody comes in with new onset diabetes, diagnosed on a blood test, the sugar is high, and then, okay, you have diabetes, and then the clinician will go to the electronic medical record where the sequence exists. I think the sequence will be able to tell you this is a subtype of diabetes you have based on the clusters. Maybe it tells you measure a C-peptide, measure adiponectin, and then the two things combined give you overall risk. And then if there is some demonstration from our work that having that information leads to a different therapeutic choice, then the computer will tell you, and for this subtype of diabetes, you need a GLP-1 receptor agonist, or you need metformin plus this. And so I think that's the way it would happen. It's very nice to be able to work with clinical trials where we can do the genetics and explore, as we are doing in GRADE, and we're going to present this at the ADA and the ASD on how that matters. But, that doesn't have SGLT2s, etc. But, there are companies that are interested in this. They don't want to segment their market necessarily, but if the genetics helps them for better efficacy or to avoid side effects, I think that's something that will be helpful.
Great having you in Copenhagen, Jose. And I think what you were showing us sort of also speaks to why variant to functional analyses, especially for type 2 diabetes and maybe also other traits, are so complicated, because some variants will act on beta cells under certain circumstances, others act in the liver cell and in liver cells. Some might even act in the brain, muscle cell, and so on. So I wonder if you didn't talk much about the sort of variant to function work, where you, based on a variant and a relevant model, piece by piece, are sort of stitching together the pathway that which in a given cell population may lead to some dysfunction that ultimately manifests in type 2 diabetes.
Yep.
But I think what you told us is sort of a good example why it's complicated. It's you will need so many different models, and you will need to know where a given variant act, and then at some point build it all together. And that seems to be an effort that can't be done in a single group, maybe not even a single center, but maybe sort of calls for a consortium that sort of just makes those cellular models and then works on trying to synthesize all this information into some sort of model that at some point could be used to model the etiology of type 2 diabetes, at least the etiology called by genetics, in a somewhat unified setup.
Yeah, so that's true, but I don't think that impedes clinical translation. So let me just, which was why I was asked to speak about clinical translation. So variant of function is key. You know, the fact that you have an allele in a GWAS that changes in frequency between disease and health, and then you can state that statistically, still doesn't tell you what is the effector transcript of that allele changes, how does that effector transcript or regulation of that effector transcript lead to changes in the cell, and then how does that lead to changes in the organism. That is, very, as you mentioned, very laborious variant of function work that can only happen one variant at a time, and it requires collaboration. And I'm glad that CBMR and Broad, our sister NNF sponsor centers, that are engaged in precisely that kind of activity. But you don't need to know about mechanism to ask what is the predictive effect of a variant on either disease outcome or response to therapy or development of complications. So to give you another example from medicine, we used aspirin to prevent heart attack because we knew it worked, not because we knew how it worked. We knew that if you give aspirin to people with pre-established coronary disease, you prevent the next heart attack. Now, I can tell you that, it's cyclooxygenase inhibition, and it's affecting platelets, and it's doing all these things, well, maybe, but that did not need to happen to know that if you give people aspirin, you prevent heart attack. So I think knowing genetics, how it affects human phenotypes, and how it affects response is something that you can do without necessarily having to understand mechanisms. So I think both lines of work can proceed in parallel, but one is not necessary for the other. It's helpful, it's nice to understand, but as long as I know that it works empirically, that's all you need.
Yeah. Thanks, Jose. So extending on Tune's question probably is, so without having to know function though, but we have to choose things to work on in pharma, right? So we have a target list, a GWAS list. What levels of genetic evidence convince you that a target is worth investing in to understand function, right? Or to understand whether it's a true pharmacological target? Is it the statistical p-value? Is it the odds ratio? Is it loss of function variance? What are the levels of evidence for you? Mendelian randomization, or what is a top-ranked piece of evidence where you go, yeah, that's why we need to work on that?
Well, I think the very first thing you need is you need to make sure that you know what the effector transcript is of that signal. So, many of the variants, the great majority of the variants are falling in non-coding regions. And, you don't know whether something nearby or distant that has been affected by that. So the trip from genetic association to really identifying which is the actual SNP that matters, meaning what is the allele that when changed, say, experimentally in CRISPR, leads to a change in expression somewhere else. So from the original index SNP to the causal SNP, that's some work. Then from the causal SNP to the effector transcript, which is the gene that is affected by this being changed, that is a lot of work. And then once you know what the gene is, then is it in tissues that make sense for my phenotype? And is it being expressed at a time when an adult intervention makes sense? Because this is about beta cell development, and week 3 of development is not going to be therapeutically accessible. So I think there's all that needs to happen. So you need to be quite convinced that you understand what gene you're talking about as the causal gene, what tissues is working on, and is more or less druggable at an appropriate time. And then I think at that point you pursue it as a target. So GWAS is only unveiling the very first thing, and there's all this extra work that needs to happen. And I think it happens best in a collaboration between industry and academia. And I think industry brings in the perspective that we don't think about as to what is druggable, and of course the landscape is very crowded for type 2 diabetes therapeutics when you have drugs that are so effective and so good. And so at some point, the discussion of what is value added for the therapeutic landscape also needs to happen. Well, was that all one before GLP-1?
So maybe give the mic.
I mean, that was what you did at Eli Lilly and Novo before you developed GLP-1, yes?
No. I mean, I think the genetics gives you, for the first time, it looks like the human is the organism that you can experiment on, because nature has experimented and it gives us, we have the tools now to be able to assay that. And so all the preclinical work, not necessarily helpful, because as you know, there's so many things that die when you go from preclinical to clinical based on side effects. So I think the ability to use Mendelian randomization to generate positive evidential proof that manipulating this pathway matters for human phenotypes, and the ability to use GWAS to say, actually, I don't see any signal for untoward off-target side effects. It's a great shortcut for, I think, companies to be able to use this information to make go, no-go decisions when it comes to therapeutic programs.
We'll have one more question in the top.
Thanks. David from Copenhagen. Could you speak a little bit more about what needs to be done in the future to better these techniques? And what I mean by that is, should we be focusing on more specific phenotypes? There was Ralph DeFranzo was on the Peter Attia podcast recently, where he was like, we need to clamp individuals and see, is this muscle or fat or liver insulin resistance, so that we can better GWAS these phenotypes? Or do we need to work on better methodologies from the genetic side, whereas we know some of these illnesses or diseases are 80% hereditary, but these GWASs and these PRSs only capture 5 to 10% of the overall variation when we compare it to sibling studies.
So yeah, the problem with very refined phenotyping like clamping is that you can't do that in enough people to generate power. So when we're first designing GWASs and trying to understand the genetic architecture for insulin resistance, we had to make a decision. Do we go to the most sophisticated phenotype available, which is the M component on an insulin clamp, which we have maybe a few hundred clamps available in the world with GWAS data? Or do we go to HOMA-IR, which is a very crude, dirty method of insulin resistance, only using fasting glucose and insulin, where we have 40,000 people? And so you can actually make a calculation in terms of your statistical power based on the correlation between HOMA-IR and the M component, which is about 40%, it's not great. But then you're changing sample size from 40,000 to, maybe 2,000. And so it turns out that at that time, it made more sense to do a GWAS for HOMA-IR, because you overcome the limitations of the dirty phenotype by your very much larger sample size. I think now that we have 50% of the heritability explained, and we have these polygenic scores that go with subtypes of diabetes that are process specific, and therefore you explain a greater proportion of the genetic variants, now a 1,000 clamps are actually helpful. You can actually detect differences by putting a polygenic score in 1,000 people with clamp data. So I think for discovery, it's still better to use large populations, UK Biobank, etc., and the biobanks like that for discovery. When it comes to then phenotypic characterization of what you've discovered, we are now at the point where the aggregation of alleles into polygenic score and the number of clamps that are available is helpful. So I don't think they're mutually exclusive, but I would use discovery in large populations first, and then characterization and refined. Yeah.
Perfect. Thank you so much, Jose. Let's give an applause again.